This episode of the Curious About Cannabis podcast is brought to you by you, our dedicated listeners and supporters. Thanks to your continued listening, sharing, and donations, we've been able to continue the show free from third-party advertisers and sponsors. So, thank you. And if you'd like to learn about other ways you can support the show, visit patreon.com/curiousaboutcannabis.
Hi, my name is Dr. Arno Hazekamp. I'm from the Netherlands, and I have been a cannabis researcher in the lab since 2001.
The Curious About Cannabis podcast is produced by Natural Learning Enterprises, a mission-driven company dedicated to enhancing critical thinking skills and public scientific literacy about life in the natural world. If you like Curious About Cannabis, consider checking out some of these other learning initiatives by Natural Learning Enterprises.
Come on, Molly! It'll be an adventure! Phoebe called out as she followed Brother Toadstool. Brother Toadstool led Phoebe and Molly into a tunnel that went deep down into the ground. As they climbed into the tunnel, they found themselves getting smaller and smaller and smaller.
Our new children's book, A Toadstool's Treasures, takes young readers on an adventure into the fun and fascinating world of fungi. Learn more and find mycology-related learning resources, games, and lesson plans for teachers and homeschooling families at toadstoolstreasures.com. Biodiversity loss due to habitat loss and fragmentation is rapidly increasing around the world with devastating consequences. Learn how you can help contribute to native habitat corridors in your community and reconnect with your wild neighbors at gardenwild.org. Oregon recently became the first state in the United States to legalize the medical use of psilocybin. As cities all over the country begin to decriminalize the use of entheogenic plants and fungi, it's time to have a serious discussion about psychedelics. The Serious About Psychedelics limited series podcast is coming soon. Learn more at seriousaboutpsychedelics.com. You can learn more about Natural Learning Enterprises at naturaledu.com. And now, back to the show. Hey everybody, this is Jason Wilson with the Curious About Cannabis podcast. Thanks so much for tuning in once again. So today I am really stoked to finally be crossing paths with somebody whose work I've been studying for quite a long time and have wanted to talk to for ages. I'm here with Dr. Arno Hazekamp. Arno, thanks so much for being willing to come on the podcast today.
Hey, Jason. Well, I guess we're both curious about cannabis, so I like doing interviews and talk about this topic, so I'm happy you asked.
Absolutely, yeah, and it's always fun talking to fellow scientists, and one of the things that I'm interested to pick your brain about later in the conversation are some of the unique experiences that we have as scientists working in this interesting field. And you've been at it quite a long time.
Yeah, that's true. I started in 2001, and we don't just do the work in the lab, we also just employees. We work for different companies and different bosses, so we also get to see the cannabis industry from non-scientific angles. So I'm very excited to talk to you about all these different things in the coming hour or so. But yeah, I've been at it for a while. I always tell people I was at the right time at the right place, or the wrong place and the wrong time, whether depending on how you look at it. Yeah, indeed. I was a biology student studying molecular biology. I thought it was the most amazing thing in the world until I realized that if you study it, that's one thing, but you probably also will have a job in it when you're done. And so when I got to the end of my studies, I suddenly realized, like, wait a minute, I don't want to work in this field. I just want to know how it, how it sticks together, and what conclusions you can take from all these exciting experiments. But to do it for a job, for a boss, for money, that's a different thing. So I took a little break that summer, and I looked around and I thought, okay, what do I care about? What do I want to find in my future job? And I decided it needs to be not just the lab part that I really enjoyed as part of my molecular biology education, but I also wanted to have a component that was socially relevant, because
Yeah.
my problem was that I thought I was doing great things in the lab with microbes and genes and PCR techniques and pipettes, but nobody understood why. That I was a
Yeah.
genetic manipulator or a Frankenstein, and it was for fun. I know that, but even my mom didn't have the patience to really understand what I was doing day to day, and I realized I missed that. I want to have a
Yeah.
job where people want to hear, what they can understand when they have, even if it's a minimal amount, but they have some kind of an interest in it, or they know something about it. So after this long summer break, I ended up in another department where they studied the chemistry of medicinal plants, in a nice word that's called pharmacognosy. Some people actually know that word, and some people think I made it up. But that's what it's called, and it's the study of plants as a source of new medication. So you can imagine that after a few years in that field, working on hops for beer and on biofuel from algae and on healthier tomatoes and all kinds of chemicals extracted from plants, cannabis came up. So one day my professor came into the lab and stood in front of me and said, "Hey, Arno, you're done with your master's, looking for a PhD. You want to combine lab research with some social relevant aspects. So what about medicinal cannabis?" So I looked at him with big eyes and I said, "But why? What is this? Where'd you get that?" He said, "Well, the Dutch government has decided that they want to legalize cannabis as a medicine. You can already go to Dutch coffee shops and buy whatever you want there. And our current health minister doesn't think that it's responsible to do that. And she also recognizes that there's quite a few people doing that. So there's an actual health risk and there's an actual gain to be made. So she wants to set up a program with a cultivator, a laboratory, a distributor, packager, all these different things. And so we need a scientist. So how about you?" So Holland and Canada became the first two countries that really organized this thing top to bottom. And I've been at it ever since.
Wow. And before we totally dive into the cannabis stuff, because I can't let you off the hook after mentioning some of your other work, what were some of the things that you were studying around hops?
Well, that's interesting because, people sometimes think that if something has been studied or not, it's a very black and white, right? We've studied
Right.
it, so we
Yeah.
know. We have not studied it, so we don't know. But the interesting thing, sometimes we've been doing things for a long, long time, and we still don't actually know how it works. And the
Mhm.
funny thing about hops at that time is that there were 6 components. They're called iso-alpha acids. They're a little bit related. Of course, hops and cannabis are a little bit related. So these are also terpene-based compounds, and there's a bunch of 6 that may or may not contribute to the taste of beer. They are
Okay.
very
Gotcha.
important. We know that because in hop extract that's used for beer, they're present. But they scientists had never been able to isolate them or to even identify them in a chromatography lab. They couldn't be separated, so they were all bunched together as one. But
Mhm.
what if 5 of them were amazing for taste and one of them was really bad?
Right.
So the Heineken company, which is actually located quite near my house, like 5 kilometers
Conveniently.
away. Yeah,
Yeah.
right. They wanted to know, what if we take one out? What if we add one more? Does that do miraculous things for the taste, for the color, for the stability of beer? So it was our task to try to find out, how do you pull them apart and how do you create reasonable amounts for initially taste tests.
Yeah.
And I did not figure it out. It was actually one of my colleagues. He worked with cyclodextrins, which was
Mhm.
a exciting way of changing the solubility of a different kind of compounds, and by changing the solubility of those 6 components in a slightly different way, he was able to precipitate them in different amounts, and,
Ah.
and that's how he fixed the problem. I do not know how the taste test ended, and I never saw any products with modified iso-alpha acid components, so I think that it was a dead-end road. But the
Mhm.
challenge for the lab is great.
And it translates very well into some of the cannabis work, because cannabis has some of these same problems where you have different isomers of different terpenes and things, and we don't necessarily know what's doing what or what's contributing significantly to certain smells versus others. The example I
Oh,
often
yeah.
use is like limonene.
Yeah.
There are different forms of limonene, and one smells kind of piney, and one smells actually more like citrus. And So that's fascinating that work really does directly tie into, I imagine, because you focused on studying terpenes in
Yeah, well, the
cannabis.
thing, with terpenes, I think for cannabinoids, the problem is not so great anymore because we've learned a lot and there's a lot of new techniques and standards available. But the problem with terpenes is there's
Yeah.
thousands and thousands of them. And they all have, well, not all, but they have mostly the same molecular weight.
Hmm.
So it's really difficult to identify them, even with something as modern as GCMS, with mass spectrometry, because not only their mass is the same, but also the fragmentation is the same. And then if you don't do a good separation, it's hard to say what's what. And I noticed that when I did some of my studies and I tried to make a clear list of all the terpenes I could identify in all the Bedrocan varieties, so that's the
Hmm.
Dutch cannabis grower that I worked with for about 10 years. And I found out that what I identify as specific terpene, other authors that were also very respected in their fields, they named them different. Not all of them, but some of them we didn't agree on, and some of them that they saw was a lot of, I saw none of it in 6 different varieties. So it still remains to be seen who is right and who is wrong, but it all comes down to agreeing, and agreeing means standardization. So
Yep.
who gets to make the final choice? And even if we're wrong, at least we're all wrong in the same way. So that still makes it better
Yep.
than what we're doing right now.
At, at least we can talk the same vocabulary and compare data.
Yeah, true.
We
But
work the same
indeed,
way.
with hemp, with hops, it was definitely 20 years ago. It was the same kind of questions. So I always say whatever we do with cannabis, it's not rocket science, and it's not new, and it's not unique.
Mhm.
It's just a combination of factors that makes it tough. But
Yep.
if the full force of science would have been released on cannabis 20 years ago, then there would have been no more mysteries by now. So we're kind of lucky we still have the mysteries.
It's true. Yeah, that's definitely true. It's like a double-edged sword. That research got so delayed.
Well, I find it hard sometimes to explain to my clients, my consulting clients, but also the people I work with in academic settings, like, why does nobody agree? Like, why can't somebody figure out a method that works? And then I have to tell them, well, I think a lot of the methods work, but
Hmm.
they just don't agree with the hundreds of steps that you have to take from beginning to end. Everybody makes
Yeah.
different choices,
Yeah.
and that's, it would be as strange as saying that when you have an analog scale and a digital scale, and one of them says a kilo and the other one says a pound, that would be strange, and nobody would accept that. And a liter
Right.
is a liter wherever you buy it. So why can't 1% of THC just be always be 1%? And that is a tough one to explain to non-lab people.
Yeah.
Sometimes even to lab people, I've noticed.
Yeah, I mean, there's so many factors that go into this. I mean, even if you had starting material that had been treated exactly the same, there are in the way that you prepare a sample in the lab can affect. When you're looking at things in such minor concentrations, it can really change things up. And then, like you said, the technology you use.
Yeah.
One thing that I've been suspicious about out here in the United States is so many commercial cannabis testing labs offer terpene testing as a service,
Hmm.
but all these labs are using different methods, different technologies. A lot of them these days are using Headspace GC, and usually Headspace GC with a mass spec detector. But I have a lot of doubts over the how valid a lot of terpene data is that's floating around, because there are so many variables, and it takes a lot of work to
Yeah,
do
for sure.
terpene testing right.
Yeah. And that's not unique in the pharmaceutical industry. There's a
Yeah.
lot of testing going on that is really complex and can go many different ways. So your results may not be the same from lab to lab, but
Hmm.
that's why there's somebody in charge making the standardized methods. That's one
Hmm.
thing. And then parties accept that standard coming from
Yeah.
the US Pharmacopoeia or the European Pharmacopoeia or other standards. And then the second thing is that you go in and you check if they
Yeah.
actually do it, and that's called auditing. And I think both parts are not developed very well in the cannabis industry because the business came before scientists and regulators caught up, and then everybody already had bought their machine and invested in
Yep.
their own favorite method. So now to make it similar, nobody wants to collaborate because first of all, everything is intellectual property, so you're not going to share anything with your competitors, and the regulatory inspection system with audits with people who actually know what they're doing when they come to check you also doesn't exist, because the doors are always closed. Nobody's supposed to know anything. Everything is covered with NDAs, and that really has to change. So it's not just choosing the methods for the many things we want to do with cannabis, but we also have to allow inspections.
Yep.
And that means
Absolutely.
and at a government level, people have to be knowledgeable enough to not just know how you do it right, but also how you can cheat, right?
Yep.
So and I can see a chance now, because the cannabis industry is shedding quite a bunch of people lately, because it's the market is not as large and unstoppable as people are thinking. So a lot of former cannabis people, scientists, workers, they go back to the market, and I think governments should snatch up some of those folks and then use them as auditors for this new industry.
Absolutely, and that's actually a very common practice. Like, some of my background is specifically in quality management, internal auditing, and external auditing. And
It's true.
that's a lot of times that's what happens in natural products labs, environmental labs. Someone gets tired of the commercial work, and then a lot of times they leave and become auditors or whatever, because they've worked on the bench, they've worked in all these areas, and so now they're able to apply that understanding in a
Yep.
way to hold people accountable. So I totally agree. Yep. And when your research with cannabis, when you first started working with VedroCan, what did that early research look like? Because I know it evolved quite a lot.
Well, just imagine that you are hired to become a technician for Formula One racing cars. So you show up at work the first day and you're like, okay, give me a car, I'm going to fix it. And you go into the workshop and there's no tools. And then they say, oops, forgot to tell you, there is no tools. You got to make your tools first. So here's a workshop for tools. And then actually we don't have the raw materials, like the wood and the steel. So can you go and get that yourself too, please? So that's kind of what happened when I started to do my PhD, because we had a beautiful academic lab with like, not just mine, but we had 10 floors of pharmaceutical research labs. So, and I knew a lot of people and professors. So if we didn't have the equipment, I could use it somewhere else. But the thing was, there was no choices made. There was only the idea of let's grow cannabis and give it to patients, but nobody had agreed on what method are we going to use, where do the standards come from, how do we package it, how do we, is there any shelf life known, what materials are even resistant to cannabis resin and stuff like that. So how are patients supposed to use this stuff? Well, we don't like smoking, so of course in the medical system you'll never promote smoking, but then what? Well, make tea or vaporize it. Well, what do we know about tea? What do we know about vaporizing? Well, nothing, but it's better than smoking, right? So I said, no, not if you're drinking disgusting green water with no active ingredients. You're just wasting your expensive medicine. Yeah, well, that's true too. So I had to go completely to the basis. So that means isolate my own standards because I couldn't order them, then quantify my own standards. Purifying is one thing, chromatography, the second thing is to quantify them. And you cannot easily do that by weighing because they're sticky and syrupy. So I had to develop an NMR method to quantify it with an internal standard that was not a cannabinoid. Then we had our cannabinoids, and I had to learn how do they behave, what kind of spectroscopic and chromatographic behavior do they have. So I had to summarize that. And then I had to move on to what's actually different between the varieties that we're growing here in Holland. Are they really that different, or should they be more different? CBD wasn't even a question back then. That came even later. But then you move on to administration forms. How do you make optimal cannabis tea in a reproducible way where it's not suddenly way too strong when you forget about the boiling water for 5 minutes because the phone rings? What if in those 5 minutes your tea gets 10 times stronger? Then there's all kinds of patients around Holland that are suddenly fainting and falling off the stairs because they're super high, right? So, and we knew none of those things. Vaporizers, what's a good vaporizer, what's a not so good vaporizer, and how would you know? No standards for anything, no quality checks for anything. If you could make a vaporizer and bring it to the market, you just sold it, right? It's not really that much different now. But then those things were new, and we had no idea what kind of toxins or crap they were spitting out, and what it even did to the cannabinoids. So my question was always, keep it simple, stick to the things that real people need to know now. No large academic studies just for your science buddies, but focus on what patients need to know today, and try to set up the study and also write the results down in a article in such a way that everybody can understand, including the
Yes.
patient you're doing it for. So
That's critical.
I call that National Geographic style, right?
Mhm.
You can't water down the science so much that it's not actually true anymore. It just sounds easy and nice, but it's not actually true, because then you're misleading people.
Yeah.
And I think, if you get data from people, you have to give it back to them. So I was interacting a lot with users and prescribers and patient organizations. Like, what are you dealing with? What does your life look like once you get that jar with cannabis and you go home? Then what? Where do you get stuck? What do you want to know? And that's been a very fruitful approach to my PhD thesis, because then not only a bunch of scientists around the world, but basically everybody in my sphere of influence, from physicians to patients to politicians to family members, they wanted to know how this works, because I can actually read this thesis and know what it means. So with that, I reached my goal of not just as a molecular biology, just doing very fun stuff in the lab, but also to have one leg in the real world and
Yep.
and be understood.
That's so important and gets overlooked so often in academia, and it's something that I've been speaking to a lot lately, because it's very frustrating as an educator to recognize how much good information is out there, really excellent papers, really excellent books, but they're written for their own colleagues, not for a general public. And so then you often have people that try to take that and distill it, but like you're saying, oftentimes it gets oversimplified to the point that it's not accurate anymore. And so I think it's really on us as scientists to take that responsibility when we write, to try to find ways to keep things
Yep.
accurate and concise and valuable to your colleagues, but also valuable to the public at large. And it's a big problem that we have, as far as our scientific literacy rates around the world are not great. And a large part of that, I think, is because of this gap between scientists and the public at large.
Yeah, I would agree. You can see that now during the whole COVID period, that everybody thinks they're a scientist, but then when they cite what they think they know, it's not totally correct. But who am I as a scientist to correct that? Because if you do it too forcefully, then you come across as arrogant or a know-it-all. And one of the topics in cannabis where I think that played the biggest role is the use of gamma radiation to sterilize the
Mm-hmm.
bioburden of cannabis. So any cannabis you grow, there's always going to be bugs on them. It's
Mm-hmm.
impossible to grow it completely sterile. And there's numbers for that. There's maximum numbers. And if you go over that, then your batch cannot be approved. So you have to do something. And gamma radiation was the only method that theoretically worked. But in practice, it's true that, that you could do more research into alternatives, at least to see if things have changed over the last 10 years or so, because a lot of patients don't like gamma radiation. They think of
Right.
the Hulk and Spider-Man and mutations and scary stuff and glow in the dark. And you can explain to them that gamma radiation doesn't do that, but it's so part of what people think that you can't change that opinion. So what I did is I was the first one to say, well, if gamma radiation does destroy all the cannabinoids and the terpenes, which is what you often hear, then you can measure that. So
Yep.
give me some the same cannabis from the same batches, and then half of it goes in a bag into the gamma radiation facility like it always goes, and the other half does not, and then I just compare. But I was
Yeah.
also smart enough to know that if I only do some complex chemical tests, people may not believe it. So I wanted to have microscope pictures. I wanted people sniff the cannabis and tell me if there was any difference. And so it appeals
Yeah.
to the scientist and it appeals to the general public, so that hopefully it's such a convincing story that they say, oh, we thought that we knew something about
Mhm.
gamma radiation and cannabis, and we may still not like it. That's your opinion. You're free to have that feeling about it, but it does not destroy cannabinoids and terpenes. That's the end of the story. And if you think it does, then you have two options. Either you show where I'm wrong, but I wrote it down in a very complete way, so at least I'm not trying to keep anything obscure. So point out where I have to do better, or you as a scientist or an organization or LP or whoever, you do a study and you show it is bad. But then we have something to talk about. And then what often happens is that such publications they put an end to how far the discussion can go, right? It's like signposts. So if you stay within the posts, then it's scientifically shown that something's going on or not going on. And if you go outside that zone, you're free to go. But then I say that's the twilight zone. That's for you. That's your opinion, and that's great. But I'm not going to follow you there, because we're going to stay inside this zone now where we have figured stuff out. And I found it very, very powerful. And I always compare that to the Mythbusters. They do that on TV. They're not judging the story like, well, that's crazy or that's totally amazing. No, they say, well, let's see. Right? So you have a whole episode to sit back and watch them do stuff and figure it out. And then when they draw a conclusion, it's really hard to say, well, I just don't agree with the conclusion. It's a stupid conclusion, because you just saw 20 minutes on TV how they went about to say it's black or white or even gray. But then how gray? You can measure stuff. So put a number on but then put a number on those discussions, make it measurable, but that only works if people agree with the measurement. I think that's the key.
Yeah, absolutely. And I mean, you're highlighting the critical aspect of science, is that we work within these bounds so that we have common points of reference, so that we can compare and so that we can be challenged. Like you said, if you disagree, then come up with a better study design, figure out some gaps in the variables I was measuring, whatever, and let's find ways to measure more things so that we can further the discussion.
But there's one problem with cannabis, because usually that's right, and standards will come floating to the top. I sometimes compare it to Betamax and VHS, right? It's not always the best technical standard. It also has to be practical, affordable, etc., etc. There's a little bit of lobbying involved. But what you see now is that because cannabis is such a different topic, it's kind of cool. It's kind of complex. It's a bit scary. It's just a high level topic. So what you see is that there is now interest in standardization, but there's a whole bunch of standardization institutes around the world. So, and I call that now the battle of the standardization institutes, because now all of them suddenly want to jump in cannabis. And now you have a battle between them. Like, I'm going to save cannabis. No, I'm going to do it first. Well, I already have a lot of SOPs. Well, I'm already working on this international something discussion panel. So especially the last year or so, from the WHO to the UN to the FDA to the European Pharmacopoeia, suddenly everybody wants to jump and then say, okay, let's standardize. But now it's just the same discussion at a higher level, because they still pick different things and different proof and different experts. So I think we're not done yet.
I totally agree. We've got to standardize the standardizers.
Yes. Yeah. No, indeed. It's the same game, but at a higher level with more experienced players. And so until then, and I think the best way to see it is looking at the CBD industry. Everybody's doing whatever they want, and sometimes it's not even legal, but somewhere else it probably is legal. So you can always point somewhere else and say, well, they're doing it. And as a result, we have 1,000 products with 1,000 different compositions, and half of those products don't even contain what they claim.
No. I mean, 90 to 95% of them wouldn't meet good manufacturing practices, whether in the U.S. or European GMP standards. It's I don't know. I think a lot of people in the industry think that when we talk about issues around product quality and standardization, that maybe we're blowing it out of proportion, because there's a presumption that cannabis is so safe that maybe those things don't matter as much.
That's
true.
I have one great example of Simpson Oil. So Rick Simpson, a Canadian who had to flee to Croatia, I think, because otherwise he would be arrested under the cannabis laws at that time. So he became a doctor, even though he's not a medical doctor, and he invented the concentrated extract with THC, which is called Simpson Oil. So I did a bunch of tests on what is in Simpson Oil, because people talk about it and nobody seems to know what's in it, but it's miraculous. So it's miraculous, but nobody knows what's in it. Isn't that strange? Don't you want to know? Everybody said, yeah, of course I want to know, but how do I do that? Well, you work with a lab, and I have one. So let me test a bunch of my own Simpson Oil recipes that I found on the internet, but also take a few from actual patients, because otherwise they tell me that I'm doing it wrong in my lab and everybody else is doing it right. And I found a ton of residual solvents in the way you make Simpson oil, which is theoretically very understandable. And I said, some of these solvents, because normal patients in their regular homes don't have access to laboratory grade super clean solvents, so they may use the cheaper ones that are available. And I've heard some stories that they actually do. So they're taking really nasty solvents like heptane and pentane and stuff like that, and chloroform. And then Rick Simpson said, or one of his disciples said, well, Rick Simpson oil is so medicinal that it will instantly cure any problem that it may or may not cause because of those impurities. So basically, cannabis is so medicinal, it can't do any wrong, right? If you just add a drop of cannabis to anything, it neutralizes it. So, and it's not really much different now with CBD. It's true,
Yeah.
and I don't want to have an opinion about CBD oil or other products without knowing. So I did my own study, but I also saw lots of studies in other places. And of course, when you are one of the first people to publish on that topic, they always ask your opinion. So I'm reviewing these papers. I'm in some panels to discuss the results, like the BBC in England. And you see in every study done everywhere around the world, at least half the products, there's a major problem. Some of them contain no CBD at all. So that means you're either very clumsy and incapable, or you're just around, a straight cheat. And I think in both situations, you should not mess with sick people. Go find another job. And I don't think that cannabis products should be over-regulated. I don't want it to become only a pharmaceutical product that only large pharmaceutical companies can deal with. I think that cannabis deserves better than that.
Yeah.
But that means that the industry has to do better, even if it's not mandatory, because otherwise they're creating their own problems. And I think that's one of the reasons why there's going to be a discussion if CBD should become a narcotic in December. So, and I think that's just a hard reset for an industry out of control that's not very willing to learn.
And to back up just a second, because I think a lot of our listeners in the United States are not aware that that's a discussion happening in other countries. So can you elaborate just a little bit more on what you just mentioned, that CBD may be deemed a narcotic soon?
Yeah, I'm not so much into it, and I am also just looking at some news on my LinkedIn, and sometimes I get an email about it, and it's not my core business. But if I'm saying this correctly, at the UN level, there's going to be a discussion in December on multiple cannabis-related topics like rescheduling, making it available for medicine, etc. But one of them is about CBD. How do we regulate CBD, and preferably in the same way all around the world? And one of the 5 topics that are going to be voted on is to make CBD a scheduled compound. Just put it on a narcotic control so that everybody has to stop doing whatever they're doing, which probably means that governments have full control over CBD again, and they can allow specific groups, specific companies and organizations to bring it to the market, but then the whole process has to start again. I think that the CBD industry has gone so much out of control because now it's available for pets, for people with cancer, for young people with epilepsy, for people who are dying, for everyone. And it never hurts. It can only do good. And that is, of course, clear nonsense because it can do bad. Not maybe because of the CBD itself, but because the other stuff that is in there as well, even up to the point of added synthetic hard drugs, because then at least your CBD product does something, right? If you want to sleep, I'll make you sleep. These things are really happening.
They're finding fentanyl in some of these products.
Oh, yes. Yeah. Synthetic cannabinoids and all kinds of things, residual solvents, polyaromatic carbohydrates, hydrocarbons, and heavy metals. It's a little bit of this and a little bit of that, but altogether, this is an industry that wants to grow really bad and really badly, really quick, and they just won't be stopped. And I can see that if I look at the kind of people that I meet, sometimes they say, well, in my previous life, I sold tennis balls from China, but that's not a great market anymore. So now I'm going to go into CBD oil. I want to help the kids, and that is sometimes so shocking that I think something does need to be done. And I don't want cannabis to be overregulated because of its history and because of the appeal to regular people, and you can actually be involved with your own medicine, but it's the business aspect, the billionaire investors and the Microsoft people, and it's become just a trading vehicle. A lot of the people that are now in the cannabis industry, they don't care about cannabis. It's just something to trade. And that, I think that's reaching its limits. You can see that on the stock markets. The stock market value of a lot of these companies is going down. They've become too big, too quick. They can't manage it, and the market isn't actually there. And even if the market is there, and you have 10,000 competitors, then you have a bigger cake but a smaller piece. And I think so. We're learning a lot of lessons, and standardization discussions and quality standards are there for a reason, but also that needs to be explained to consumers. Like, what are we trying to fix here? What's the problem, and why is that a problem for you?
Yes, yeah. And it's an extension of a problem that has already existed for, and some people hearing me talk about this are like, oh God, he's about to go on his natural products industry rant again. But this is a problem that already exists in the natural products industry, and that we've been trying to overcome with dietary supplements and other things. That it's great to empower people to have access to herbal extracts and things to handle their health however they want, but those products need to be of a known quality. They need to be of a known purity. I mean, unfortunately, we can't trust people to
It's human
watch out.
nature. Right?
Yeah.
And the same happens with cars. There's this whole
Yep.
Volkswagen scandal, and a lot of other car makers too. They put software in there so that you're being cheated on the performance of the car. So it's not just related to
Yeah.
herbal medicines or pharma
Yep.
products or anything. But I heard an example from the Food and Drug Administration here in Holland, and they said, this is not unique to cannabis. This happens all the time. Remember when goji berries
Mm-hmm.
didn't exist, and then suddenly they did, and everybody wanted them because they were superfoods? That means that somebody in Nepal, where they are grown, thought, hey, my goji berries go from 1 cent a kilo to much more. I
Mm-hmm.
need to grow more. So what we saw was a huge increase in pesticides in goji berries. So now we have a superfood, but because of the market, because of the demand, you can't afford to lose one single berry anymore because it's worth a lot of money for those people who grow it. So pesticides. So this is just how humans work. This is how business works, and it's nobody's particular fault, I'm sure. But you just need to put up barriers and therefore check systems, which involves labs, to make the invisible visible. That's how I always explain. A lab makes invisible stuff visible. You can't taste heavy metals, you can't see THC, and you can't smell pesticides. There's nothing to see. You need a lab to tell you what's what, and otherwise things go unnoticed.
Yep.
And I've dealt with patients who said that they're sleeping better than they've ever done in their life because of their CBD product, and it turns out that there's no CBD. And then when I go look with them, like, but what did you do? Where did your CBD go? It turns out that they're just not really good at calculations. So they diluted their product 1,000 times too much or something crazy. So there is no CBD. You made it this way. Oh, but I sleep really well. So how does that work? So there's a whole edge of placebo effect of wanting something to happen, and it's still a little bit exciting, it's still a little bit naughty in some places, and that adds to the myth. So in that aspect, I don't think cannabis resembles anything else today. And we just have to learn how to be more normal about it and more responsible.
Yeah, absolutely. And that is the really hard thing to tease out, especially with cannabis and how popular it is, and how big the mythos is around it, is how do you distinguish these placebo effects, specifically given the lack of clinical research with herbal cannabis or cannabis products that people are typically using, which transitions into something I wanted to talk to you about, because you've authored several papers where you've reviewed the current sort of landscape of clinical research.
Yeah, it's time for another one.
It is. It is. Yeah, I think your last one went up to like 2014. So yeah, it's time to renew. But what did you learn when you dove into all of these studies to try to see, well, what human clinical data actually exists for cannabis? What did you learn through that process?
Well, it's not only CBD producers or consumers that do clumsy things or draw conclusions that are not supported by their own
Yeah.
observations. Because it happened once doesn't mean it's a rule. But what I learned is that a lot of clinical trials are done in a pretty sloppy way too. They're too small. They don't address the real problem. They're using cannabis that nobody else can use, and it's not even medicinal grade. There's the famous example in the US, of course, is you can only use NIDA supplied cannabis,
Yep.
which is very weak, not very potent, and it's very dry. And some people have compared it to pig food. I've been in one conference of Patients Out of Time where they took one of those jars. There's only 4 patients, maybe less now, in the whole US that are alive and are using that material. And they were brave enough to bring their new sealed canister to the conference and open it in front of cameras. And it's horrible. So, and that is the only stuff you can use in clinical trials. So whatever clinical trial you do, it's not going to find what the person on the street has told you that it works for. So this is something that it's hard to change. You can't smoke in clinical trials because it's going to be really hard to defend. But I'm always looking for what can you do? Who can do this stuff? So one of the fun things I've done when I was working for Bedrocan, and I heard a clinical researcher from Switzerland complain that he wanted to do a study in Switzerland, but the only legal source he could use was NIDA because that's the only government sanctioned supply in the world. I said, but I can make that for you. What do you want? He said, well, I need specifically what they provide because that's what I've used before, so I can't suddenly change. So I need exactly 7% THC in pre-rolled cigarettes of a certain weight. I said, I can do that. What if I just take a strong cannabis and a weaker cannabis and I mix them until they're 7%? Right. So I did that for him and he was extremely happy. He said, yes, this is more realistic cannabis. It's more moist. It has terpenes. This is what people would want to use if they're patients. And actually, it's Bedrocan material, so actual people are already using it. So you have to, and it's another lesson of where scientists can forget that they're part of the real world and that they can put a lot of effort in doing the study because then they score another publication, but they don't help anyone because they just clinically proven that cannabis does not work for chronic pain. If you're a regulator, that's what you see. This is the only study on smoked cannabis and pain, and it says no. And then the scientist said, yeah, but excuse me, there's a footnote says that this was not no, don't want to hear that. It says no. So it's scientifically proven cannabis does not work for chronic pain, so stop whining about it. Those are the real world consequences, and I think you can do better. So that's why I worked on the Volcano vaporizer as a medical device, on creating a placebo cannabis for clinical studies to bring different varieties into clinical trials to compare with each other, a little bit sativa, a little bit indica, a little bit CBD, a little bit THC. Because scientists often think, yeah, there's cannabis and then you just test it, right? Cannabis is cannabis.
Cannabis is cannabis, yeah. What's the big deal?
Yeah. I think there's not as much nuance as some people like to believe, that there's thousands and thousands of unique strains, but there's also not just one. There's a bunch, and we need to know how much. And that's when, maybe you can cluster them and you can give them a letter or a number, cluster A, cluster B. But, we need to tidy this up because nobody is benefiting from 10,000 different strains. It's too complex.
Yeah, and it's very confusing and potentially detrimental to consumers, especially medical focused, patients that are looking for relief, having all of these names and descriptions when really there might not be that much difference between a large percentage. Which this relates to one of the other big topics I wanted to talk about, which is the concept of chemovar. So a lot of your some of your work focused on, like you said early on, you were looking at these cannabis materials, seeing how they were different, and eventually your research started to move into the area of, okay, the cannabinoids are roughly the same, but there's still differences. Maybe it's the terpenes. So can you speak a little bit to that research, how that evolved, and where this concept of a chemovar came about?
Yeah, actually, it was a very fun project to show how nobody needs to lose, right? It's not the scientists are right or the people who have used it for 20 years are right, and the scientists don't know anything. This was a great way to show everybody has a piece of the puzzle. So what I realized that everybody talks about varieties, and even Bedrocan had varieties, a little bit more of this, a little bit less of that. But my question was, well, how do you define a variety? Because if we have 5 products and people say, well, your selection is not complete. You need a sixth one. Then what is going to be that sixth one? Based on what? Based on what we think is cool that year, or based on CBD that just happens to be popular? But we already have some CBD. So if you have a weaker CBD strain, why don't you just use more? Why would we have to create a new product with more CBD? Because that's the same as saying, well, I don't like 100 milligram tablets of aspirin because I need to take 2. I want to have 1 of 200, and that's completely different. Then everybody will roll their eyes and say, no, they're not. You just have to swallow twice instead of once, but it's the same. So, but how do you do that when there's potentially many, many different components? So I decided to start with something that everybody seems to agree on, that there's sativa and there's indica. That is like the highest level of distinction in cannabis. Like if you have to make one cut in the whole group, you'll say sativa indica. That's for real. That's for sure. That exists. And I thought, well, if that's true, once again, you must be able to measure something. And if it's like in a compound that is 0.01% versus 0.02, then I don't believe that can be the one. That must be such a potent compound, then we would have known something about that compound by now. So it must be in the cannabinoids and or the terpenes. That's the only way. That explains 95% of what we know about cannabis right now. So what if I go out and I buy, grow as many sativa and indica extremes as I can? So there's the coffee shops in Holland, which I had access to. There's the Bedrocan collection, both of products that they had already in the pharmacy and things that were under development. And there's all kinds of other scientific connections and research licensed organizations that I know. So I was able to get 460 different samples, and I analyzed them all for the complete cannabinoid and terpene profile. Even if I didn't know what terpene it was, I just call it number 1, number 2, number 16. Doesn't matter, right? Because number 16 is always number 16 and number 21 is always number 21. And if there's not a difference, then I don't know, have to know what they are, because they're apparently not important. So why am I wasting my time? So you basically subtract things. You look at the sativas and the indicas, look at the cannabinoids, no difference. And that was interesting because a lot of people said at that time that it must be the CBD in the indica, because CBD is relaxing and indica is relaxing too. So therefore, without ever having seen the inside of our lab or having done any test, it's super logical that CBD must be higher in indica, and therefore THC is probably lower. So everybody had an explanation, but nobody had really actually put it to the test. So when I did that, I saw no difference, because they're both drug cultivars. So humans want it to be high, and high we will become, because we pumped them up to about 15 to 20% THC, and the CBD got lost in the process. So, but when I looked at the terpenes, of course, you're looking for individual components, and you say, well, if this component is there, humulene, terpinolene, limonene, whatever you want, if that's there, it must be a sativa or an indica. But I couldn't see that pattern. I'm not just looking at peaks and trying to draw conclusions. I was using statistical programs called principal component analysis or cluster analysis, and that's a very powerful tool to show patterns in huge data sets. So don't forget that I had 460 samples times 44 peaks, sometimes even more. So that's tens of thousands of data points, and you can't project it on the wall and hope you see a pattern. So the statistics told me there's certain specific terpenes associated with the indica type, but not just 1 or 2, it was a bunch. And actually, after I came back from lunch, I stared at my paper with terpenes again, and I realized, wait a minute, all these terpenes, they're ending with ol. That means they're alcoholic terpenes. Cineol and terpineol and linolol and stuff like that. So I thought, wait a minute, does that mean that the hydroxylated forms of these terpenes are linked to indica? But where do they come from? Is that an enzymatic thing? Is that an environmental thing, that where they first are formed and then they're oxidated? So does one of them have more antioxidants than the other? And then I realized that these compounds aren't made in isolation, they're made in pathways. So that means that maybe an entire branch of this pathway, controlled by maybe 1 or 2 genes or precursors, are on or off. So I was looking at individual components, but I had to look at branches, right? So that's when I realized, like, okay, so if there are hydroxylated terpenes linked, they're much more present in the indica type. Why does the plant look different? What the hell does it have to do with the plant shape? So I looked on and I realized, I found that same day that there's a particular type of plant hormones, gibberellic acids. They are made from terpenes. So either
Yep.
they compete for the same precursors, or they're boosting each other. But that's when I found for the first time a chemical functional link between the chemical profile of the terpenes, and therefore smell and taste and effect, modulating effect on the cannabinoids, versus what it does to the plant shape, the stretching of the stems, and the skinniness or the fatness of the flower, etc. And I was not able, my lab was not capable of testing and analyzing those gibberellic acids, because they are present in minute amounts and you need to be very specialized. So I never pursued that, but I think that's where the link is. So now, even though it's complex, you can still measure a complex pattern, and you can still say, well, this falls into the indica or the sativa grouping. But the explanation, you basically need to have a show and tell for about 10 minutes before people say, oh, I think I just did that 5 to 10 minutes. But it's not simple, but it doesn't mean you can't do it. But that's when it becomes science, and that's when it becomes a lab job. And I think that in the future, hopefully, instead of saying there's 1,000 different varieties, so just start with number one, and we'll see you when you hit number 1,000, and then you can tell me which one you like. I think based on this new knowledge, you can group them without any hard borders saying this is group A, this is group B. You have to do some negotiation and some talking about it. But wouldn't it be nice if you have 10 main clusters? One mostly for pain, one mostly for sleep, and another one mostly for MS. Then everybody knows what they're selling, and everybody knows much, much clearer what they're using. So that's when we went from cultivar to chemovar.
Well, and that's such an interesting proposal that the plant hormones are competing for aspects of these biosynthetic pathways that might be resulting in these alcohol terpenes. And it's hard to talk about this stuff sometimes to broad audiences, because you sort of have to go in a weird spiral where you first say, okay, the way you think about indica and sativa is not exactly right, in that the terms are used inconsistently and you may not be able to trust them. And then people get mad at you. And then you say, but there are patterns. There are biochemical patterns.
Well, what I found is a powerful way to build that bridge again is large groups of people. They know how to observe. If 100 people tell me they saw something happened, they saw a UFO, I'm sure there was something in the sky. But when they explain it with the knowledge and the tools they have, they're often wrong, because they use whatever they know, what they happen to know. Sometimes that's an old book about cannabis from the 70s or something they read on the internet. So I'm always trying to tell them your observation is correct. Same with sativa indica. It's an actual thing. But what you thought it was, it's more interesting than that. Shall I tell you what it really is? And I told you that nobody has to lose, and it was actually proven in the Dutch system, because Bedrocan was asked by patient organizations, there's always a few patients who are more vocal, and they know what they're talking about, and they know what they're trying to say, but they just don't have the scientific vocabulary. And they said, you have only sativa strains, and that makes me hyper. They're uppers, they're energizers, and I want to sleep. I want to calm down. I have ADHD. I have sleeping problems. So your products can't work for me. So the thing is that in words, they would never be able to tell me what I needed to develop next. And I don't want them to just bring in a strain from the coffee shop and say, here, grow this, because that's not scientific enough for us. So when we realized that the difference between sativa and indica can be mapped, what we did is we took those samples, the wide variety of samples that we now have access to, and we knew what they were because we had background information about the street name and the supplier and the seeds and stuff like that. So we mapped the existing Bedrocan varieties on that plot. So it's a two-dimensional plot with all kinds of dots on it, and all those dots were the different types that we measured. And then we plotted the Bedrocan strains on them, and we saw that there were two major regions that were like on a map. We were not occupying them with a product. There was nothing there to fill that gap. One of those clusters was a mess. It was all kinds of stuff mixed up. But the other one was all amnesia haze samples, because they happen to be very popular in Dutch coffee shops. So you see a lot of different types of amnesia haze from different sources, and they were not occupied. Well, that amnesia haze was a indica type of variety that we had access to. So we picked one of the plants that we had already in our collection, and we grew that to be the next product. And then there was a very exciting moment where we presented that product to that group of patients and say, "Is this what you asked for?" And they said, "Wow, yeah, that's it."
Wow, nice.
So they were right by just smelling and sniffing and tasting and trying and smoking and talking about it. They knew what was, where they wanted to go, and through science we could put numbers on it. And then in the end, those two totally came together. So that was very exciting, and then you can share the whole story with the patients. The patients were, I think, even allowed to name the new variety,
nice.
or at least to be involved in that. So they helped. They really helped, and it was one of the best presentations I ever gave at a conference, because that was like literally half, like 1 foot in society and 1 foot in the lab, and then you come up with one solution. So it's a great example.
That's exactly what I was going to say. It's a beautiful example of how science and the public can work together to enhance each other's work. Trying to get on a similar footing as far as the vocabulary you're using, and trying to keep an open mind to really understand and hear each other.
Well, another example is raw juicing. I remember raw juicing became popular, and I happened to be in California around that time because I had my girlfriend was there for study. So I heard about raw juicing, and of course it was immediately associated with healthy hippies from California. Of course, they want to juice everything, and it has to be raw because then you live longer and there's no toxins. But so my colleagues were laughing about that, like, "Oh yeah, great, and it tastes horrible, so you have to add all kinds of superfoods. So one smoothie costs $25." But what I heard as a chemist was raw juicing, raw smoothie, raw, no heating. These people are not smoking, they're not vaporizing, they're not baking any cookies, they're not making any heated oils, they're eating it raw. So they're eating the acidic cannabinoids.
Yep.
That is interesting, because nobody does that. So why are they doing that? They're not doing it for the taste, I'm sure, and not because they actually want to be super happy with the minerals and the enzymes or whatever. But even if they do, wouldn't it be cool if you find out that all the raw juice drinkers, let's say 90% of them has a specific disease like irritable bowel syndrome? Then have you just discovered in the wild, like an anthropologist could do in the rainforest of Brazil, you made a link between irritable bowel syndrome and THC acid.
Right.
Well, that is worth getting out of the lab for, and looking around with your chemical X-ray glasses to see what are people, not what are they talking about, but what is actually chemically happening. And I found that if you go around the world in that way, then a lot of crazy stories are true, and a lot of true sounding stories are crazy, and then we're back at the Mythbusters. Because some very plausible things turn out to be totally impossible, and the other way around. And that's what makes this fun. And I think that's how you should employ a lab.
Yeah, exactly. And part of what you're describing is something I talk about a lot here with the concept of curious about cannabis. It's about questions, and it's about when you have that kind of perspective, you're able to identify better questions to ask, which I think is really one of the keys to science and understanding. Is you start out sometimes asking ridiculous questions because you've got to start somewhere. And as you learn and share information, your questions get better and better and better until you start to really touch at the core concepts that you didn't originally understand.
Yeah, one good example is clinical trials. A lot of people in the world are kicking and screaming that there must be clinical trials, because otherwise, we are completely unhinged and we don't know what they're doing. It could be dangerous. But clinical trials are difficult. They're expensive. They, usually the results are owned by someone that doesn't want to just share it with everyone. And my idea is, well, if we want clinical trials, but we just can't have them, period, not right now, it's going to take years. Why don't we do the second best thing? And why don't we ask tons and tons of people really smart questions in a survey, which is already being done around the world, but there's always one thing missing, and that's because There's a bunch of scientists, they're doing the surveys, but they don't touch the cannabis supply. And there's other researchers around the world. I know one in Israel, for example. They look at the samples that are going around, but they never talk to the people. And what if you can talk to 10,000 or even maybe 100,000 people that are consuming cannabis for health? And at the end of this really good survey, you say, okay, can I now have a sample of whatever you are using?
Yeah.
I don't care if it's illegal or not. That's not my job. I'm a scientist, I'm not the police. And I know this is hard to do in real life, but there's always places, countries, conditions that you can do it. So now I have your answers about what it does and what it doesn't do to you. Now can I link it to your chemical profile in the product that you're using? And I don't care where you get it from this supplier, that supplier, you grow it yourself, because I'm just finding the pattern. And I can guarantee if you do that, just like with the sativa and the indica, you'll find a pattern, and you'll understand why certain components are linked with a better outcome for MS, sleep, cancer, or irritable bowel syndrome. And then you can switch it around and say, well, from all the hundreds of possibilities that we could have studied and wasted a lot of time and money, now we have narrowed it down to a few probably winning combinations. So now let's get that proof.
Yep.
And you don't waste time and effort and because academic time is worth money too, right?
Absolutely.
You have to score, you have to publish. So you don't want to publish a clinical trial that didn't work.
Yep.
Nobody needs to see that. So narrow it down with real life data. But that means that actual scientists, this is not just a regulatory problem, that means that actual scientists have to come out of their ivory towers and interact with actual people. Those are not just your guinea pigs numbered 1 to 100. Those are people with knowledge. They know something you don't know, and you know something that they don't know. So now let's work together. And that's a kind of a cultural gap that I found is difficult to force. You need some time.
Yeah, it's a nuanced thing. It takes some finesse to really work out. But you're absolutely right, and something that my mentor and I used to talk about a lot in the lab was the concept of tribal knowledge. That that's something that is worthwhile to tap into, understand, record, document, and not just out in the like among users, but even in the lab. Like, we have tribal knowledge as scientists in our heads and things that sometimes you've got to tease out from your colleagues, and it's important. And to loop back around to the raw juicing thing. So first of all, did you do chemical analysis on the juice to see what was actually being ingested?
No, at that time I got excited and I talked to other people about it, but there was a little period of my life that I didn't have direct access to a lab because of I was with my partner, and she was focused on her for about a year. So so I kind of passed on that topic to another, but yeah, of course, the acidic cannabinoids came into the spotlight around that time, and it means that there's more people looking at more interactions, because terminology can be very powerful. If you say that THC is the active ingredient, and who wants to study the precursor? Who cares about the precursor? Because I'm going to smoke it up, or I'm going to put it in a cookie. So nobody cares about THC acid. That's just the precursor. So, the non-usefulness is already part of the name. It's a precursor to something that really cool, which is the active ingredient. So I always balked against that kind of terminology, as there's just a bunch of chemicals. They could do something or nothing. But there's also the other end of the scale. People would come to me at conference and say, Arno, what does this cannabinoid do? What does this cannabinoid do? And I would say, well, I don't know, maybe nothing. And then they looked at me completely incredulous, like, how do you mean it does nothing? It's like, why does it all have to do something? If you believe that, that's great, but that's called intelligent design. Then the cannabis plant is chosen and it's stuffed full of cannabinoids, and they all do something, and they all cure horrible diseases. And only now we find out. And other plants are just plants with chemicals, and they do nothing. Right? If you eat a horse flower, I'm not going to ask you, what does this compound do? It just tastes bad. That's the only thing it does. So, and that was the time, I think, that science wasn't doing much, but consumers, and there weren't that many producers yet, but the cannabis world went a little bit overboard. Everything had to do something, and had to cure something, and had to be put into something, and it can never harm. And I think that cannabinoids may indeed not do that much harm, but can you imagine that cannabinoids are positive for cancer in any way or form, but there's one type of breast cancer where cannabinoids suppress the immune system and you die faster. Wouldn't you want to know?
Right.
Or not every judgment you have about cannabis or cannabinoids or anything you measure is a judgment call, right? So that's
Yeah.
why I always show in my presentations a picture of the Mythbusters, and I say these guys are not there to judge; they're there to just figure out what is plausible, what's not plausible. So if some cannabinoid doesn't do anything, or maybe actually promoting cancer in one way or another, doesn't mean that we're trying to bash cannabis,
Yes.
right? And I
Yeah.
think we're now starting to come in the decade of cannabis where we can do that a little bit more unbiased on
Yeah.
both sides, on all sides.
Yeah, it's something that has been striking to me. Someone that I interviewed some time back is a person that suffers from cannabinoid hyperemesis syndrome,
Yeah, that's
and
one of them.
so
Yeah.
they share their story, and they've become a very big advocate to try to raise awareness about it. And she gets a lot of hate from the industry
Yeah.
under the assumption that if she's talking about a negative aspect of cannabis, then she must be secretly anti-cannabis, or
Oh, yes.
trying to influence people to think negatively about the plant. And so this ties into another thing that I want to talk to you about, because in your education work that you do
Mm-hmm.
a lot these days, is you focus on myths around cannabis, and all of these perceptions that the culture kind of juggles. And so one thing I wanted to ask you are, what are some of the most prevalent myths about cannabis today that you frequently encounter?
Well, let's just search my brain a little bit, but one that comes immediately to the top is that it seems to be quite well accepted that different labs have different results because they're different, right?
Hmm.
So of
Hmm.
course it's different results. So you just pick the one you like, and then what's the problem with that? I said, well, can you imagine going to the gas station and then filling your car up with 20 gallons or whatever, how much goes into a car these days, and then it's not 20, but it's 16? You would be
Right.
mad. What if you
Yep.
go buy a computer and it promises you all kinds of cool things and speed and chips and memory and stuff, and it turns out to be not true? Wouldn't you be pissed? Yeah, of course you would be pissed. What if you buy a bottle of wine and it's not 12%, but for all these years it will happen to be 9? They just failed to tell you. Then even if you didn't get less drunk or nothing bad happens, you're going to be pissed because you're fooled as a consumer. So why is it so strange that for cannabis and cannabis products, you want to demand a label that just is correct? I always say the simplest thing you can say about cannabis products is if it's on the label, it must be in the product. And if it's in the product, it must be on the label. Why? Because it's normal. You
Yeah.
don't have to have any deep discussions about what's true and what's not true in conspiracy theories. Doesn't anybody want that for everything? Because if it's not, then you just don't know what you're doing anymore. So let's start there. And it's confusing when people believe that because I have a more modern machine, you are using HPLC with UV and I'm using MS detection, and mine is hipper and cooler, and I'm a university and you're not, so my numbers must be more correct. And that's nonsense.
Yeah.
More complicated equipment needs more dedicated and skilled use as well. You can do more things wrong. Maybe your standards are not really what they're supposed to be because there's some confusion there. So it's not okay if people start to think that labs are different because they just invested in different directions. If whether you have an analog scale or a digital scale, and if you buy digital scales from 10 places, they all have to say that a kilo is a kilo. So and that is a difficult discussion to have. And I find that if you use analogies and examples and move away from cannabis, and you try to show people that it's not just about cannabis, it's about how the world works, about bigger structures, and about what what you can actually test and what you cannot. Some things are just really complicated. uh, yeah, that's worth a discussion.
It
Another thing,
is. Yeah.
if you think about microbe testing, are you doing it with plate testing and then you only
Mhm.
test the living microbes, or are you doing it with PCR and then you may also measure the dead microbes? And what's the difference and why does it matter? And then of course the added problem, especially in markets that have been a little bit older, if you tell Dutch people that cannabis in coffee shops may be dangerous, so we need to have a higher quality, they're going to say that, but if it's so dangerous, then why the coffee shops exist? It's
Mm-hmm.
they're here for 45 years. So if
Yeah.
it's so dangerous, then why didn't anybody die? Then why didn't I hear about it? So how subtle is that with heavy metals and pesticides and mycotoxins? It's very gradual scale, right? It's
Yep.
not dead or alive. So if you undertell that story, people get a false sense of security. But if you overdo it, people start to see conspiracy theories, and it's going to kick you in the butt. So you have to do it right, and it means you have to do it very calm and very gradual, and don't go too quick. And that's the
Yeah.
role of scientists, I think.
Yeah, no, that's a really good point. What you just highlighted is things like pesticides, mycotoxins, whatever, and metals. These are things that typically accumulate in the body over time. And you're right, they may not kill you, but they can influence your health in all sorts of ways, and can negatively impact your quality of life over, your lifespan, possibly make you more susceptible to certain diseases, possibly promote things like neurodegeneration, at a faster rate than normal.
But I think that even if they want that, that's up to
Hmm.
them. I'm not
Yeah,
deciding for them. So if
sure.
it's there
Yeah,
and it's on the label, and you say, well, this is a more expensive brand because it's super clean, and this one is not so clean
yeah,
because it has craps in it and residual solvents. If you want to buy it, fine. And my job as a scientist is done, but you have to understand the choice. And the rest is up to the regulators, of course. But if you don't measure, you're making rules for categories that may or may not exist. You're
yep,
just doing it blind. I always say if you don't have a lab, it's like you're trying to judge colors, but you're colorblind.
yeah,
And you're trying
yeah,
to imagine what those colors are going to be like, but that's not really accurately possible.
yeah, yeah. No, absolutely. Another thing that I wanted to talk about that just popped in my mind that I know you studied, that I forgot to ask you early on in the conversation. So going back to cannabis tea, I
Mm.
know that you published a paper specifically looking at cannabis tea, because it was such a common preparation. Um, what did you learn about the chemical profile of a cannabis tea? Because there's a lot of debate over how effective a cannabis tea might be, given that cannabinoids are not particularly water soluble.
Yep,
Yeah,
indeed.
can you expand on that?
Well, the first thing I learned is cannabis tea can make you extremely high because I was in the lab making, I think I was focusing on 1 liter portions because we were
Mm,
teaching people to make enough for a while. So you don't have to do it every single time, but you can store it. But I was taking little 30 milliliter samples for the freeze dryer to do the final testing. And when I was pouring it down the sink for the 12th time or so, I thought, isn't it weird that I'm studying it, but I have no idea what it tastes or what it does? So at the end of the workday, I decided to have 1 little cup of cannabis tea, and 2 hours later it hit me like a tsunami. I was from 0 to high in literally 10 seconds. It was
oh wow.
unbelievable. It was very fun and a little bit scary.
Yeah,
um,
yeah.
but that kind of taught me a lesson that if you give cannabis to patients, and that's new, a lot of patients are not users and they think that a tea is a mild form of using it because they're not smoking it. Like, is
Mhm.
that actually true? Yes, the cannabinoids are not water soluble, but the recommendation based on theoretical assumption by the Ministry of Health was take 1 gram of cannabis, put it in 1 liter of, so about a quart of boiling water, and boil it for 15 minutes and then filter
Ah,
out the cannabis. So I
okay,
thought, well, at
okay.
least they thought about heating and stuff, but is 15 minutes in boiling water, is that hot for a cannabinoid? Well, it turns out no. Cannabinoids don't care that much. That's not very hot. That's why vaporizers, they start to really work at about 150 C or more. But 100 degrees doesn't do much except blow off the terpenes. So it smells great,
Right.
but what's in the tea? So I thought, well, what if I'm told 15 minutes, then that's the baseline. Then I follow that recipe. I have a number. But what if I don't have a precise scale in my
Mm-hmm.
kitchen? So
Yep.
1.00 gram is not very doable at home. It may be anything between zero point 0.5 and 1.5 before the scale turns from 1 to 2 or from 0 to 1. What if I boiling for 15 minutes and the phone rings? My mom's on the phone and I
Mm-hmm.
forget about the time. So what does that do with the composition of the tea? So my focus was not so much how do I make it as potent as possible. We are trying to actually protect naive patients from too many cannabinoids. The purpose was to make it robust.
Mm-hmm.
So I did 5, 10, 15 minutes boiling the water, half a gram, 1 gram, 1.5 gram. So I did all kinds of different parameters, and I found out that by coincidence, actually the recipe of the government was pretty robust.
Hmm.
If you boil it shorter, it becomes less potent. If you boil it longer, it doesn't become more potent. It either doesn't become more potent when you add more cannabis, because it turns out that you get a saturated solution.
Yeah. Mhm.
So it stops. But if you put it in the fridge, then everything starts to stick to the glass, and within a day it's just water. And this is relevant because I had heard about a research group in the University of Groningen, which is in the north of Holland. They were MS specialists, and they were the first one to be brave enough to say, we're going to study this in a clinical trial setting. So they made a whole bunch of tea,
Oh boy.
stored it in the fridge, and gave a bunch of patients, I think it was 6 patients, they gave cannabis tea for 5 days. And they said, well, it doesn't do anything, so we've proven it didn't work. And I was the only one with the knowledge that I had. I said, well, but how much tea did you make? Well, we made 5 liter. Where did you store it? In the fridge, right? They said, yeah, because that's recommended. So did you add anything like milk powder or like milk to keep things in suspension? And they said, no, because nobody said that. I said, well, was there a little sticky circle on the glass just above the level of the water, of the tea? They said, yeah, but we rinsed that out at the end. I said, yeah, but that's not the point. That's your active ingredient. So, and you could not know that because nobody knew that, but you have
Yeah.
to add something, even just a simple like milk powder that you use for your like coffee creamer powder, because it's easy. Everybody has it. It's super stable, and that works. So my recommendation was follow the recommendations, but if you make more, add a spoon of coffee creamer to keep it in suspension. Otherwise, after one day, you've just wasted your medicine. And then a lot of people may conclude, I tried cannabis, and the only way that I find it acceptable, I do not want to inhale. I tried it as a tea, and it didn't work, so now I'm moving on, and I will not
Yeah.
use cannabis anymore. And that's a waste.
Yeah.
That was the motivation for this study. Like, let's keep people in the cannabis medicine world, without making them disappointed, but they don't even know why it failed.
Right.
Let's take out these obvious mistakes.
Well, and getting back to what you were saying before of like making sure people actually have a choice. So giving them good data so that the choices they make are solid. They're based on reality and not just a failed, procedure
Yep.
or something like that. And it's interesting what you're pointing out, because I think when we first started talking about cannabis tea, probably what most people may have thought about was boiling some water and then pouring it into a cup with a tea bag of cannabis in there. And so your research highlighted that if you followed the recommendations that the government had set of boiling it for you said 15 minutes, is that right?
Yes. Yeah.
15 minutes, boiling it for 15 minutes, that's good. But maybe what people would do without thinking about those recommendations of just putting cannabis in a tea bag and pouring hot water over it and letting it seep for 3 minutes, that may not be good enough.
That won't work. Yeah. It was also interesting that tea contains way more THC acid than THC, but the solubility of THC acid and the other acid depends on the pH. So maybe it depends on if you add other stuff. Some people like honey, some people like to add rooibos tea or something. So it's really hard to know what people are going to do at home. And I think that's a major problem with medicinal cannabis. The way it was distributed then is that you tell people to do something, but are they going to do it? What if you tell them to not smoke it, but they smoke it anyway? Or you tell them to make tea, but they do it their special way? So, you have to know something about it. So yes, of course, when you make tea, you're losing some of your active ingredients and your decarboxylation could have been better. But it's a trade-off with the fact that it's a very robust and self-limiting concentration because it gets saturated. So that means that people can't do it right 10 times in a row and then they forget about the time and then they're suddenly knocked off their feet because they're high, because getting high for a lot of chronic patients is super scary.
Yeah.
Losing your mind like that and having the feeling that your chest is going to explode. That's what happened with me with the cannabis tea incident. With all the knowledge I had about cannabis, I still thought that maybe I'll get a heart attack because my heart was pounding like insane. So this just happens, and I think it's good to have that experience sometimes, and sometimes the scares on a completely different level. For example, I am using CBD from Holland, where you can easily get it everywhere in very great looking stores. Just it's an official product, and I go on vacation to Eastern Europe or to Russia. This actually happened. One person went to Croatia, other one went to Russia. They do a drug test because they don't really know what CBD oil is, and it sounds kind of cannabisy, and it tests positive for THC. Is there THC in this product? Yes, there is, ma'am. There's a little bit. That's totally legal in Holland, yes, but I'm now in a Russian jail, and I'm being accused of drug smuggling. Couldn't you have told me before that this contains a trace of THC? Yeah, but the Dutch law doesn't allow that if it's below a certain level.
Wow.
So, harm can come in a lot of different ways.
Yeah, well, and that's a really good point to bring up, and I think we would be remiss if we didn't specifically talk about this. What, other than some of the things that we just talked about, what are some of the real risks and harms associated with cannabis that you think people should have in their mind? Without exaggerating or trying to freak people out, but there are some real things to make sure people are aware of.
Yeah, well, of course, one is potency. If you think you're
Yeah.
buying 10%, but it's actually 2%, then your product instantly became 5 times more expensive. So I'm scammed as a consumer. I lost money. That's not great. Second thing is maybe there's nothing in there, which happens quite a lot. So I think I'm curing myself or treating myself, but I'm actually doing nothing. And what if that's cancer? And I so trust in cannabinoids that I choose not to have chemo, and this can be a huge a drama in a family setting. When your family members, please take this chemotherapy because it's 60% effective. So no, I trust cannabinoids, and then you're not actually using cannabinoids. What then? Or you're stuffing yourself with cannabinoids and it doesn't work. It doesn't do anything, and it actually suppresses your immune system. So maybe you sleep better and you eat better, but you're still dying twice as fast. How would you know? That's why you do clinical trials. Another thing is that people can spend a lot of money on these kind of treatments, and they think it's holistic and better, but it's not covered by health insurance. So I can also see that people lose money on that. People who are not in the position to lose that much money. And then there's the contaminations, anything from some residual solvents and heavy metals to the purposefully added hard drugs that are found. And this is not an incident anymore, I think. This is happening quite a bit around the world, anything from synthetic cannabinoids to powerful modern medicines, because then at least you have an effect from that product, so that product will become popular. And then the problems with THC contamination. It's not just that you may get stuck in Russia, and then when you're lucky, in this case, they sent this woman back after two weeks, but she was there on vacation for family business and for actually running a company. And then what if you're told, sorry, you're not welcome for the next 2 years?
Right.
That's a big price to pay for something you didn't choose purposefully. And what if you're using CBD products for your epileptic kid and it turns out to be full of THC? Maybe not last time and maybe not this time, but then suddenly next time there is a lot of THC. And this has all been tested in places like Australia and in the US, Canada, Germany, Czech Republic. It's happening everywhere, UK. Every time there's a publication about CBD and contaminations, you find stuff. And I think that has to stop.
Yeah, absolutely. It's really challenging to even assess the efficacy of cannabis when some of these variables are at play.
I have two nice examples of companies. I was doing a little bit of myth busting for about a year because it was getting out of control in Holland just as anywhere else. There was one company which is a tiny little pharmaceutical company right next to my town, so I never heard of them, and they claimed that now they had discovered that CBD can come from orange peels. So we don't need to. There's another story about hops. So to make a long story short, I asked them, how does this work? Can't tell you. There's a patent. It's from a German company, so we can't say. Okay, let me guess. You're taking some terpene, like limonene or something, from an industrial source, which indeed may come from orange peel, and you're making synthetic CBD. Is that what you're doing? And then they shuffled around a little bit, and then they ignored my email, but I kept pushing, and I said, yeah, you're kind of on the right track. I said, but you said you extract it from orange peel. And they didn't think that was misleading. That was just kind of a way to communicate about this. But 2 weeks later, this advertisement was gone. So no orange peel at all. And the same story has been brought up by hops and other things. And people can really get scammed for years. I think the whole hop story about the special strain of hops from the Himalayas, it lasted 2 years. People invested millions in that company. It's ridiculous. But that's the damage you can do. And the other one said, well, I now have a product that is 50 times better, 52. He was very specific. The bioavailability, the uptake in the intestines is 52 times higher than any other product. And I've proven that in studies. So my product is worth your money. And so I called him too. And I said, well, how did you do that? Because I cannot see you with your background and everything I can find. You cannot do clinical trials. So where is this done? So I also dug deeper and asked more questions. And it turned out that they had done a study on 3 isolated mouse intestines. So not even in vivo, but with isolated organs. And there they found an elevated penetration of their CBD. So I said, so you didn't do it at all with humans? No, but we're planning that. So, we don't know how to translate it to the market. But what we meant to say is we're on the track. I said, well, but why don't you say it? So there's misleading information everywhere. And some of it comes from labs, unfortunately. There's also labs that see an upside to reporting consistently higher numbers. So if you want high numbers, they'll give it to you. So everybody is a little bit involved, and there's forces and counter forces on every level. But as a consumer, you can't see any of that. The only thing you see is that glass jar in the store with a price tag.
And the marketing language that gets shared, all over social media in a matter of days. And it takes years to counter that.
It's kind of a challenge to take the lid off and to take people into this wild wonderland of stuff that happens in the cannabis industry without having the intention to make cannabis or the industry look bad. I'm happy that it's here, and I think cannabis presents a whole new way we can do medicine with cannabis and many other psychedelic plants and products maybe in the future. But I also think that what we do now sets the tone, because I'm already getting some requests, some consulting requests, and I read stories about magic mushrooms. What about their medicinal potency? What about varieties in mushrooms? What about growing it under GMP? And the same is happening with ayahuasca and other kinds of plant-based psychedelics that were fully legal. But I would not like all the mistakes of the cannabis industry see it come back in like double speed, triple speed, fast forward in these other industries, because it's going to make a few people rich and it's going to cripple the industry as a whole. And then maybe in December, the United Nations will vote CBD a new narcotic, and then we get stricter instead of more open, because we let the industry play without restrictions for about 10 years.
Yeah, and I mean, in psychedelics, you don't have the wiggle room you have with cannabis. If things go wrong, they go wrong in a big way that is very impactful in the moment.
Well, just think about it. What if I'm a CBD producer and I say, you know what, this stuff doesn't harm anyone? Just use it for everything, and it's good for kids and old people and dogs, and so there's nothing to be worried about. But at the same time, it can cure cancer and epilepsy in kids, and when you're dying, it does all kinds of great stuff. So you're saying at the same time, it doesn't do anything, just leave me alone. And at the same time, you're saying this is a medicine that works when everything else fails. So it's potent and it's harmless at the same time. That's just not possible. So I think it's okay if you market CBD, but then no claims in a very dilute form, and then you can put it in some stuff and it's like echinacea or valerian root, or it is potent and powerful, and then it has to be regulated like a medicine. You can't have it both ways.
Yeah, I mean, that's the thing. When you start getting into the world of medical claims and all of that, you open up a door into, you have to standardize, you have to know quality, you have to do clinical studies, and not even just, something that I talked about with the guest recently was you have to do studies on your active compounds and the formulation itself, but you also have to understand the non-target, the quote-unquote non-active components of the formulation, depending on how you've made it, and what do those things do, and are those things toxic, and
I think there's a very deep belief among a large group of people who like and support cannabis and that believe is cannabis can't do any harm, and that's I think a consequence of all these decades of misinformation and governments
Yeah.
trying to push it to a point where it's almost ridiculous. And now we know. So now for the next 10, 20 years, we will not trust any official source saying anything bad
Mhm.
about cannabis. It's kind of a rebound situation, right? But
Yep.
why would cannabis be always harmless and only be good for you? There's no such thing in this world. So I always found it a little bit stunning. It's almost religious. So I come always back to intelligent design. Somehow the cannabis plant is chosen and can do things that nothing else can, not even other plants. And I just think that's very unlikely. But I hope it's true. I hope cannabis cures cancer. I'm just not convinced. It is a challenge and interesting to try to lift the lid on all the stuff that's happening in the cannabis industry worldwide, and
Yeah.
showing out the things that are not working well, and the scams and the problems that are out there, without taking down cannabis as a medicine in general. I think cannabis creates great new opportunities to do clinical research, to interact with patients, to create new medicines that are much needed. But there's a whole bunch of people, organizations, and companies that are not doing so well, maybe not even
Yeah.
intentionally. But so how do you show the good and the bad at the same time? And I think that is something I'm always very aware of and try to be very cautious with.
Hmm.
So that's why sometimes when I'm with patients, I defend government standards, but when I'm with the government, I'm defending, experiences by groups of patients versus clinical trials. Why do clinical trials always need to trump everything? So I think basically what cannabis does, it takes the pipeline of medicine development, where a scientist develops something, a company patents it and makes a product, and the authorities allow you to use it in the first place. And then a physician or a prescriber allows you as a patient to use it. But you're at the mercy of all those guys and all those different roles, and you're just lucky to get it because you're sick. And
Yep.
it creates a situation where patients may think, but wait a minute, this is about me. I'm sick. Why are you making
Mhm.
all these choices for me? And cannabis, what it does, what it has done, is it just goes straight through like an icebreaker. It didn't care
Yeah.
about any of those things. It started with patients, and then everybody jumped on board. So now they have to sit around a table instead of passing things on in the pipeline. And I think human society is just not used to that.
Yeah.
Patients talking with physicians, and then the physicians actually listening to be inspired for future studies. Like, oh, wait a minute, what?
Yeah. What
or
is this brave new world we're stepping
or
into?
companies being involved in making regulations to regulate themselves. Like, wait a minute, we don't do that. So there's always a picking order. And
Yeah.
cannabis as a medicine changes that picking order. And I think it could be, if we do it well, it could last for a while and open up a
Mhm.
whole new way of finding and developing new medications.
Yeah, I totally agree, and I really appreciate your perspective and approach to this. That we're trying to understand what's real and what's true, and we're not trying to bash what people are saying. We're not trying to invalidate people's experiences with cannabis, and likewise, we're not trying to take what's published in a research paper that's been peer reviewed and say that that's the gospel, and that we ignore everything else. I think that's such an important stance, and it's one reason why I like a lot of your education work, and that you're focused in education, because that's so needed.
Yeah, education. I think one of the things you've then mentioned, or you mean, is the masterclass medicinal cannabis, which I set up in
Yeah.
2011, and it takes a whole week. It's face to face. It's a small group of selected
Mhm.
people, selected for their diversity in countries where they come from, but also their level of education, their connection with cannabis, everything from users to physicians to scientists to government people, because in such a diverse group, especially when you're stuck for a week and you have to have dinner and lunch
Mhm.
together, stuff comes out.
Yep.
And I love to see how people are maybe a little bit more combative, or they all agree in the beginning, and then it changes because they realize there's just a lot of nuance.
Yep.
Um, and that's how education becomes a little bit of like bringing up. Now, I'm not saying that, right. The thing with cannabis is you can't just tell people what you know, because they're not blank. If you tell people something that they don't know, they're going to say, oh, this guy knows a lot about it. So let's listen. I'm learning something new. And you put it in your memory and it's a new piece of the puzzle. But with cannabis, everybody, even my grandmother, even my little cousin, everybody thinks they know something about cannabis from TV, from movies, from education, propaganda, all kinds of sources, from the coffee shops. But none of those pictures is complete. So if you want to teach somebody about cannabis, you have to deconstruct something first without making them mad and offensive. And then you have to feed them the new knowledge. And it takes time. You have to let them say what they like, what they don't like, what they've observed, why they think that that's true. And only then, by exploring together, you can really learn. So half the course is taking down and the other half is building up again. And then you see that most of your building blocks are still there. They're just reshuffled a little bit. And I think that's what can give people the confidence that, okay, I'm not wrong, but yet it's different than I thought. So, right, it's about not having to lose and having to say, okay, fine, I was wrong 50 years of my life. Nobody wants that.
Yeah, exactly. And I think as an educator, taking that responsibility of facilitating the discussions in a way that helps people not think in those terms of right and wrong, but a perpetual pursuit of understanding, and your understanding can mature over time, and it should. I mean, there's an old quote, I'll mess it up, but there's an old quote that I like a lot that's like, if you've never changed your mind, then you're a fool, because I mean, you have to change your mind as you learn. Otherwise you're stuck in your original position.
New information needs to go somewhere. If it goes into the same old letterbox, then you may as well learn nothing, and then you already figured the world out when you're 12. That's unlikely.
Exactly. Yeah, exactly. Yeah, well, it's super fascinating hearing about your education work, because you and I are parallel in a lot of ways. Obviously, you've done so much work that I've not had the opportunity or been able to touch. But as far as how we approach education, and like this class that you're discussing, one of the ways that I got into doing the work I'm doing now is coming out of the lab. I put together sort of a workshop that mimicked what I wanted to see a college level survey cannabis science class look like. And it's very similar. Like my goal was to get people in person, understand why they're passionate about learning, and how to facilitate discussions and connect them with good research, a lot of your research, and how to think more. I even hesitate to use this word because people take it the wrong way, but more maturely, to think, to be able to put some of our biases aside and just talk and explore ideas and come to some new ground, a new foundation to work from.
Yeah, I don't know if it's a bad choice, maturely. It definitely rings with me. And part of that is by taking down barriers. And a great example is when I was doing my test of CBD products. So I asked 40 people that I knew and that I got to know for this project, can you send me your CBD a product, because I want to know what's in it. I want to know if it's the same or different, if there's THC or not. And as a reward, I'll give you free testing data, because that's not something you can easily get. And there were, of course, people that didn't agree with the test that I did. And they said, no, you say that my product is 1%, but I'm pretty sure it's 10 because I made it myself, or I bought it from a really reliable person. So instead of going into a discussion, it's like, you know what? Bring your sample, bring another sample, come to the lab. We're going to do it together. I'm at a university. I'm allowed to have you in the lab. Maybe I'm not 100% allowed, but I'm just not going to ask anyone. So come over. We'll do it together. You can see from beginning to end what I'll do, and I'll explain everything. I'll write everything down on a piece of paper. You can calculate with me. So I'll show you what happened. And it seriously turned out that somebody made a calculation mistake, and they diluted their product too much. And they said, oh yeah, I'm not so good at math. That's true. So the problem wasn't at all where you thought it was. So this person was much happier because now he knew how to make 10% oil, because 10 divided by 10 is 1, and I found 1. So you made a great product. You're just not good at math. So that is mature too, I think, without
absolutely,
at least politically mature. If you look at political discussions, if you're not winning, you're losing. And I think we have to get rid of that idea. If you're not above somebody, you're below it. And that has to do with the pipeline of medicine development. I'm important because I'm the regulator, and you have to follow my rules, which is true most of the time in the real world, but I think cannabis provides different ways to get to the same goal. If we want
Yeah.
clinical data, then a clinical trial is not the only way you can get there. It's just one we know very well, but what else could we do? And I find it exciting to come up with different paths to the same endpoint.
Yes, absolutely. Yeah. It's almost like being a scout or a map maker.
Yeah, and we can get attached to something. So if sativa
Yeah.
and indica wouldn't be real things, then we call them Jupiter and Saturn. Who cares? Now you're
Right. Yeah.
now you're opposing it because you like sativa and indica and it has a meaning. But in 10 years, we'll have forgotten all about that and we'll use the new terms as long as they're useful, as long as we can explain what is the purpose they serve and are they more accurate than what we've been doing so far. So
Yeah.
that means that there's a little bit give and take on all sides.
Yes. Yeah. Yeah. Absolutely. Well, I know we've been going for just close to 2 hours here, so I want to be respectful of your time and not keep you much longer. But to kind of wrap things up here, the last question that I'll pose to you, and then I'll hand the platform over to you to close out, is um, what currently in the cannabis and cannabinoid science world has you is sort of has your excitement peaked? What research are you interested to follow over the next several years? And then after you address that, I will also just hand the platform over to you to let people know how to learn more about your master classes and any of your other work, and pretty much anything else you want to share with our listeners before we sign off.
It's a big platform. Thank you.
Yes, totally.
Well, one of the things I'm involved with now is what we call the Legal Cannabis Coalition. So I worked at
Mm,
Bedrican for 10 years, and then we decided to say goodbye to each other for multiple reasons. So I was a consultant, but
mhm,
one with a large network. And it means that if I'm serving clients, and I can pass them on to a whole bunch of experts in the rest of the field. But one of the things that also happened is that I started to notice that Holland was often overlooked as a country where exciting cannabis things happened because
mhm.
we're just not that big. There's one licensed producer. Our medical program isn't giant. We don't produce tons and tons and tons of cannabis. So I realized that there's a lot of specialized companies in Holland that do everything from building greenhouses for cannabis growers to watering installations to laboratory services, making clinical trial medication and things like that. But each of them by themselves were too small to be noticed in the big scheme of things. So we decided, why don't we put them all under one umbrella and do some of our own research and also act as one group? So that would be called the Legal Cannabis Coalition. And one of the things we're doing in collaboration with the University of Wageningen, which is a very world famous agricultural university, one of the top 10 in the world, they are allowed and able to do cannabis cultivation trials. And they're
Mm,
doing it for research, fully legal, but they're not allowed to bring anything to the market. It can be sold to any single person.
mhm.
So the fun thing is, if you're not selling to the market, you can make mistakes on purpose. And any
Yes.
LP would never do that, maybe on a tiny little scale, but definitely not repeat it 3 times with controls all around. It's just too much money. It's too much time, and it doesn't do anything for your business. So we're able to look at LED lights, about different nutrients, and about different pest control methods. And one of the things that we've been doing that for almost 3 years now. So we're in our 7th cultivation round. It just finished. And one of the things that we can't get a handle on, but it appears that almost nobody in the world can get a handle on, is botrytis.
Mm,
It's
mhm.
it's a fungus that is in the air. It's always there. You can't get rid of it. It bugs a lot of other agricultural plants as well, but we don't know where it comes from and we don't know what to do about it in cannabis. And entire crops are lost. Entire greenhouses have to be burned and slashed down because of botrytis. So one of the things we're trying to do by monitoring the climate conditions, putting all kinds of probes and cameras and sensors on these flowers, um, controlling water flow, humidity. So basically, it's like a high-tech patient stuffed with sensors. But the problem is, if you have 1,000 plants, you don't know which one is going to get botrytis. Could be any, could be
Yeah.
none, could be all. So which one are you going to point your cameras on? You don't have 1,000 cameras because they're expensive. So, there's a little bit of cat and mouse game going on with the
Mhm.
botrytis. And now I have an idea that there's a key somewhere in the terpenes, because we have noticed, and I think many growers have noticed, some cultivars always get botrytis, some of them never, almost never. Why is that? Is it like immune system? And if it's immune system, then what compounds are responsible? And I'm starting to zoom in on specific terpenes. I don't know which ones and how many and what ratio, but something's going on. And I would have a dream that you can figure it out and you can create an effective and very harmless treatment for botrytis contamination in cannabis.
Fascinating, yeah, and that's has such a huge impact on so many cultivators. I mean, like you said, and so going back to this idea of practical research as well, that is super practical on so many levels.
If you connect it to earlier research, it's a good question, but I don't know, because
Yeah.
you need lots of data points, and every individual LP doesn't have enough data. But if you
Right.
combine it, do indicas or sativas have different sensitivities for botrytis? Like real extreme, not the mixed ones, because then it's hard to say.
Yeah.
But the extreme sativas and the indicas is one of the more vulnerable, because then again, maybe it translates to terpenes or maybe even growth hormones. So
Right.
and then you can
Well, and
close the loop, in a different way.
and it makes you also ask questions about the effects that breeding techniques have on the resiliency that the plants have, because there's some work's already been done that has shown that as you breed for higher THC levels, you often lose natural resistance to powdery mildew, for instance.
Yeah, could be.
And so it'd be interesting to look at it on a genetic level too. Once you kind of have a sense of what might be influencing those dynamics, then you can start to look at the enzymes, look at the genes responsible for producing
I
those
think what
things.
we're saying here, in a different way, you and me, is that we need lots of data points, which means that we need to collaborate. It means that everybody has to
Yeah.
share data, and then we put it in statistical programs in an industry that is notoriously bad at sharing anything.
Yep.
So I think that's the big data coming to cannabis, but it doesn't mean big data inside your own LP. It means big data and data patterns in the world of cannabis, not just US, not just Canada, but just everywhere. And who's going to do that, and how are you going to make that happen with all the legal restrictions and business restrictions that are in place today? So I think we still have at least 10 years work ahead of us.
Yeah.
Before it becomes standard.
Absolutely, and as we've seen, it's not a linear trajectory either. We take steps forward and back as we ride this roller coaster towards new regulatory models and new industries, and so yeah, it's going to be fascinating, and I think this all ties up our conversation really nicely, highlighting what we can do if we can put aside biases, if we can standardize some things so that we can have common reference points and be able to compare data, draw more meaningful conclusions, and ultimately just continue to do things better. I mean, from the world of quality systems, it's all about continuous improvement. How do we continue to improve as we move into the future?
But I like to emphasize it's not rocket science. And very soon in the near future, everybody can grow cannabis of a decent level, but it's going to be super cheap and you're not going to make a lot of money, and everybody can make things out of it. Making CBD oil, it's also not rocket science. So that just means what's left behind is that you have to make 100 choices. Are you going to do it this way or that way? And so there need to be good steps. It's not going to be tough, but it's going to be, you have to be responsible.
Yep, exactly. Well, awesome. Well, Arno, thanks so much for spending the past couple of hours talking to me. This was as enjoyable as I expected it to be. I mean, when we talked on the phone before doing the interview, we seemed to hit it off pretty well, and this is just a nice continuation of that. So I really appreciated the good conversation, and I hope that in the future we can reconnect and talk even more about some of this stuff.
I would hope so too.
Change.
There's lots of extra and more topics to talk about. If you want to know more about my work, I've created one website, very simple one, where everything comes together. It's www.hazekamp, my last name, hazekamp.info. And there you can find the news about my publications. A lot of them are on there, about training, about consulting. other things. So, and I love to be approached for interviews and other kinds of interactions.
Perfect. Awesome. Well, yeah, everyone listening, go check out his website. It's very well designed as well. It's got a very nice sort of simple presentation that makes it very easy to find stuff.
And you can see what I look like as a cartoon.
Exactly,
as an avatar. Yeah. That's my favorite part.
Yeah, I need to get one of those, figure out what I look like as an avatar. Well, yeah, thanks so much, everyone that's listening. Thanks so much for tuning in and listening to this conversation. If you want to learn more about Curious About Cannabis, you can go to cacpodcast.com or find us on social media. We're on Facebook, Instagram, and Twitter, primarily active on Instagram. But yeah, thanks so much. This has been a great conversation, and I look forward to connecting with you again soon.
Thank you, Jason, and great day for the audience. Hope we'll speak again.
I think so.
Okay.
Yes. All right. Take it easy, and bye-bye.
Bye-bye.
If you want to learn more about cannabis, check out the Curious About Cannabis book on Amazon.com and other major online book retailers. To support the show and get access to an exclusive members-only podcast feed, access to private events, merchandise discounts, and more, visit www.patreon.com/curiousaboutcannabis.